Today, we're going to compare gastrulation patterns in the bird, sea urchin, frog, and mammal. And we're going to examine what's taking place during neurulation, which in vertebrates is the initial phase of organogenesis. Last lecture, we talked about the 3 stages of fertilization, which are penetration and fusion of the sperm to the egg membrane, activation of the egg, including 2 forms of polyspermy blocking, and fusion of the 2 haploid genomes and formation of the diploid zygote. We also covered cleavage, and cleavage is rapid cell division that ends with the formation of the blastula. We can separate the process of development into fertilization and cleavage, which we already covered, followed by gastrulation and organogenesis. Now, gastrulation is when we're going to see the formation of the 3 tissue germ layers, ectoderm, mesoderm, and endoderm. And organogenesis, which in vertebrates begins with the notochord development and neurulation, is when the 3 germ layers of the embryo differentiate and further specialize to form the various organs of the body. And today, we're going to focus on gastrulation and the initial stages of vertebrate organogenesis. Here we're going to look at variation in blastula structure. In the mammal blastula, called a blastocyst, we see an inner cell mass at one end or pole of the structure. The outer layer, called the trophoblast, will eventually contribute to the formation of the placenta. And we also see a central fluid-filled blastocele. The sea urchin blastula is pictured here with labels indicating the 2 hemispheres, the animal pole, where smaller, more rapidly dividing cells are located, and the vegetal pole, which is where the larger yolk-containing cells are located. Now, the bird blastula is called a blastodisc, and it sits on top of the yolk. Also, recall that the bird zygote goes through meroblastic or incomplete cleavage. This is due to the increased amount of yolk found in the egg and yolk's resistance to cleavage. The frog blastula shares structures with the others and has a fairly substantial number of yolk-containing cells at the vegetal pole, but the frog egg does carry out holoblastic cleavage. Now, in gastrulation, cells change shape and move to other locations in the blastula. And this process establishes the basic body plan and creates those 3 primary germ layers, the ectoderm, the mesoderm, and the endoderm. The ectoderm being responsible for the formation of the epidermis of the skin and the nervous system. The mesoderm forms the skeleton, muscles, blood vessels, blood, gonads, and kidneys. And the endoderm forms the lining of the digestive tract, the liver, the pancreas, the thymus, and the thyroid. Now, there are 4 basic types of cell movement during gastrulation. In invagination, the cell sheet dents inward, like shown here. In involution, the cell sheets roll inward from the outside. In ingression, cells break away from the outer layer and migrate as individual cells into the blastocele space. And in delamination, we see the formation of a second sheet of cells, and those 2 sheets of cells then split in 2. They separate. Now, sea urchins develop from yolk pore eggs, and they form these hollow symmetrical blastulas. Now, sea urchins and all of the other models that we're looking at today are deuterostomes, which means the anus forms before the mouth does. And gastrulation in sea urchins involves ingression and invagination. So we can see here ingression taking place. These primary mesenchyme cells are separating from the outer layer and migrating into the blastocele space. And then here we see the formation of the blastopore through invagination. And then on this picture, we actually see the formation of the archenteron, which will eventually form the digestive tract or digestive tube of the organism. So here in this 3rd picture, we see endoderm that's forming the archenteron. We see ectoderm is the blue pictured area. And then these red dots are mesoderm that's suspended in an extracellular matrix. Frog blastula have asymmetrical yolk distribution, and even though the yolk-containing cells of the vegetal pole are less numerous, they are larger than the yolk-free cells of the animal pole. Now, in frog gastrulation, which includes invagination and involution, we see the formation of a blastopore on the equatorial region of the blastula. Now, this is followed by involution, where cell sheets move inward from the outside of the blastula, from the blastopore. And then here in the third image, we see that endoderm, which is pictured here in the yellow layer of cells, mesoderm, and ectoderm, the blue cells, are all present. In bird gastrulation, which involves delamination and ingression, the blastodisc becomes a two-layered cap of cells called the blastoderm. Then those two layers delaminate, forming a blastocele. These two layers here form a blastocele when they come apart. Then a transient structure called the primitive streak, pictured here, forms and ingression takes place as we see cells from the upper layer of the blastoderm cells break away and migrate into the blastocele space located in here. And then here, as with the other examples, we see the presence of the endoderm layer, the mesoderm layer, and the ectoderm layer. Gastrulation in mammals is similar to that in birds in that it also involves delamination and ingression. And first we see a flattening of the ICM as the amniotic cavity forms between the ICM and the pole of the blastocyst. Then the ICM's two layers delaminate, forming ectoderm and endoderm layers. The primitive streak appears, and cells break away from the ectoderm layer at the top and migrate to the interior, forming mesoderm. Now, immediately following gastrulation, organogenesis, which is the formation of the organs of what we can now refer to as an embryo, begins to take place. And in vertebrates, this initially involves the formation of the notochord and the nerve cord or neural tube. And this occurs by interactions of cells both within and between the three germ layers. And this also involves migration of cells. Now, neurulation, or the development of the neural tube, refers to the folding process in vertebrate embryos that includes the transformation of the neural plate into the neural tube. And then after this is complete, we can refer to the embryo as a neurula. And this process begins with the formation of the notochord pictured here. It forms from the mesoderm tissue and will eventually become the vertebral column. After notochord formation, the dorsal ectodermal cells located up here thicken and form the neural plate. And then the cells of the neural plate fold together to form eventually a long hollow tube. And this neural tube is the precursor to the central nervous system. It will become the brain and the spinal cord. So today we looked at the different patterns of gastrulation that we see in the sea urchin, the frog, the bird, and the mammal models. And we also discussed the process of neurulation as a major event in organogenesis. And we explored aspects of neural tube formation.